slides look good. And with that, it's my pleasure to introduce our second keynote of the workshop, Shantanu Singh. Shantanu is a senior group leader at the Broad Institute, where he co-leads the Carpenter Singh Lab, a data science group that develops computational and statistical methods to create fingerprints of genes and chemicals from microscopy images of cells to probe biological processes and finding new cures for diseases. After completing his PhD at Ohio State in computer science, Shantanu joined the imaging platform at the Broad to help realize cell morphology as computable as genomes. He has previously worked in research groups as Mercedes-Benz R&D, GE Global Research, and Lawrence Livermore, and is fundamentally excited about bringing together the many rapidly developing fields of high-dimensional biology to tackle the challenges of improving human health. Likewise, we're excited to hear him speak today about finding disease phenotypes and candidate therapeutics using cell painting. Please take it away, Shantanu.
Thank you, Stephen. Can you hear me okay? Audio good? Excellent,
yep.
Excellent. And I'm going to turn off the video just so that we can make sure there's no interruptions. Well, thank you for having me back and for the invitation to present our work. It's really exciting, really, to see this community build up momentum. Today we were here about a couple of years ago, and it's really nice to see that a lot of the ideas we discussed just 2 years ago that seemed blue sky are already being realized. So today I'll talk about how our lab at the Broad is using microscopy imaging and machine learning to address many problems in biomedicine and drug discovery. I will focus on the broad topics of how do we extract information from millions of images of cells to characterize cell state, and then show you examples of how we can use that information in biomedical research at large, and more importantly, how to use that to find new drugs for diseases. I plan to leave plenty of time at the end, at least 20, 25 minutes, so that we can have longer discussions, but please feel free to drop in questions into chat, and we can take it at the end. We are surrounded by pictures of stunning biological images anytime you walk into a gallery of science. For example, like this one at the Koch Institute at Cambridge, or works of art like these by Derek Sung at UPenn. But embedded in all this is something even more amazing. It's a rich trove of information, as many of us know, that we can use to understand and characterize diseases using bioimage analysis software. The imaging platform of the Broad is the home for one such software, Cell Profiler. It's a tool that's widely used as a workhorse for analyzing images, but I won't be talking at all about image analysis today, per se, or Cell Profiler, except to mention that the most deeply satisfying outcome for its creators, and I'm sure for the creators of any bioimage analysis software, has always been the role that it's played in finding new cures for diseases. And much of what I'll talk about today would be how our research, which is based on information extracted from software like these, can help drive discoveries that can impact patients. I want to mention our sister lab, led by Beth Samini, is entirely focused on developing the next generation of bioimage analysis software, building on the rapid progress in recent years on bringing deep learning innovations into this field. So select use of imaging, where we use tools like Cell Profiler to measure specific features or identifying specific phenotypes we care about, is impactful, but we know that there's more than a single readout or cell phenotype that we can get from an image. In fact, in the field of profiling, of image-based profiling, we argue that by collecting hundreds of readouts from pictures, just like what you're seeing on the screen right now, we can capture a surprisingly expansive view of cell state that can help us probe biology. The concept of using images as profile, as opposed to just measuring particular features, is maybe easier to understand in the context of thinking about how diseases are diagnosed. Many of us can tell a child has Down syndrome on the appearance, by the appearance of the child's face. And sometimes you have to be quite an expert to be able to discern the different diagnoses from a face, like with Engelmann, for example. But this task is something that machine learning can be used to do very effectively for many disorders. Now, you can't look at a person's face and diagnose all diseases, of course. You actually need to dig a little deeper by taking blood samples to look closely at their cell structures. But the fundamental concept is the same, right? There's something about the morphology that's telling you what's going wrong with that disorder. And if we can make that kind of diagnosis in a dish with the patient's cells, for example, we have a potential to screen drugs in order to potentially correct that disorder. So that's the basic concept of image-based profiling. But what's the best way of scaling it up? What's a good way of scaling it up in a lab, right? One way of scaling it up is to stain cells with as many dyes as you can pack into an assay. The cell painting assay uses a set of 6 different dyes that labels 8 different constituents, cell constituents that are imaged in the 5 channels shown here. These are cheap dyes, and it makes for a very high throughput and inexpensive assay that anyone can run even using a regular lab microscope, using the protocol papers that I've mentioned here to guide one along. Then, as many of you know, we can also scale it up in terms of the number of perturbations we can measure if you use automated microscopes and machinery, and then also scale it up by just extracting thousands of features from each cell, which is no surprise to you. Now, once we have all that, we now have represented each condition. That's typically a chemical or genetic perturbation or a disease state of some kind into a rich collection of single cell data. And that is a great framework, as you all know, for doing high dimensional biology, because research questions can now be formulated as essentially machine learning problems in this space. And what we're most interested in is how do we use that to dramatically impact different stages of the drug discovery pipeline as represented in the schematic. For example, starting at the top right here, can we use this approach to identify signatures of disease and then find drugs to revert them? I'll focus on just one example on this front. Together with Miko Taipala's lab in the University of Toronto, we're exploring this exact idea in the context of rare diseases. As an example, if we label a protein such as the LZTR1, it has a certain localization shown here. But if you make the mutation in that protein that causes Noonan syndrome, you see an obvious change in the protein's localization. The phenotype is so clear that we can now readily test drugs to try to make the disease cells look healthy again using automated image analysis. So we can scale that up. The Taipale lab has created an image-based profiling data set for more than 1,000 Mendelian diseases across more than 3,500 mutant alleles by overexpressing them in a cell line. The initial goal was to systematically characterize just the protein localization of the disease variant alone, and doing so by scoring images by eye. Jessica Lacoste in Miko's lab led this work. We're now taking a fully automated approach, automated and unbiased approach to this problem. That is, we're looking at not just protein localization, but a broad set of morphological measurements to find unique signatures of mutants in this work led by Marzi Hagigi in our lab. For mutants with distinct profiles, we would get some insight into what are the structural changes in the cell, but also then be able to screen for drugs that can potentially revert that phenotype. Being able to scale up drug discovery like this is great, but we can go a step further and also do all of this in a pooled optical setting, and I'll talk about that in a little bit. So moving all the way down, can we also use image-based profiles or just imaging for diagnostics? So here's an example of how we could do that to directly help patients needing a blood transfusion. So over time, blood stored in banks undergo changes that impair their health and ability to carry oxygen. The changes make the blood unusable in treatments like life-saving transfusions. Now, healthy red blood cells begin to transform from these smooth concave discs to lumpy round balls and then to swollen dense spheres. Experts currently look for these changes in morphology of blood samples under a microscope. They just count how many cells just fall into each class and then combine them to get a morphology index, which they use to grade the blood quality. The process can be obviously slow and prone to human error and subjectivity, as you imagine. Using deep learning to identify phenotypes, Juan Caicedo and Min Duan in the group and others had used just first tried to mimic this task. That is, we trained a classifier to bin cells into each of these morphologies and found that the performance on this task is pretty good with the agreement of the system to expert annotated ground truth, approaching the agreement between different experts on this task. But do we really want to do this? Because that would simply be mimicking the system designed by humans with a somewhat arbitrary phenotype bins, right? What would be really powerful to do is to get a continuous measure of each cell's quality rather than these discrete bins, and then use that to report a quality index. We did exactly this using a weakly supervised deep learning approach, we trained a neural network to predict the age of the bag to which a cell belonged. We don't really care about the age of the bag. Obviously, we always have that information, but older bags are expected to have more degraded cells than newer bags. And this information was useful in making the network learn a representation of these cells that could, in a fully automated way, get a proxy measure for each cell's quality. The measure can also be translated into an index, and it turned out that the so-called self-learned index had a much stronger correlation to the biochemical assay than the index calculated by experts. Importantly, the approach is fully automated and free from human bias, and if validated in follow-up studies, it could be used to create a new standard for assessing the quality of blood bags. Another role that profiling can play is in getting clues about the mechanism of action of potential drugs by querying their profiles against an existing library of chemicals with known mechanisms, or even genetic perturbation, because matches with those might reveal their putative targets. And we'll talk about the latter in a little bit. In fact, in this paper that came out just a month or so ago, we compared two different high-throughput profiling assays, cell painting, which reports on morphology, and L1000, which reports on the transcriptional state of about 1,000 genes. We profiled about 1,500 compounds, mostly FDA-approved drugs, across doses, and we found that across the two assays, compounds in about a third of the mechanisms present in the data set were significantly tightly clustered. In this analysis, morphology is consistently higher than L1000 in terms of the number of mechanisms captured. You can see the light blue right here. Sorry, I'm not pointing to that. The light blue in this plot being the overlap. So when you do an unsupervised analysis and just measure the similarities of profiles and report the metrics. But the story is a little bit more balanced when you do supervised analysis, when we train a classifier for this task. The two axes here report the area under the precision recall curve for the two assays. We find, not surprisingly, that some mechanisms are better predicted in one assay versus the other, so there is complementarity between them. This work was led by Greg Way, Adeni Adeboye, and Ted Natoli and others, and is our most thorough investigation to date on comparing these two assays in general, not just on the task of prediction of mechanism of action. And you can look up the paper for more details. In another paper that came out a couple of weeks ago, led by Marcia Hakiki, we provided data sets for achieving a different set of tasks involving these two assays. We've curated cell painting and L1000 data across thousands of compounds and several hundreds of genetic perturbations with the aim of being able to translate across the two assays in useful ways. Going in one direction, we can create predictive models of expression levels of individual genes given their morphology readouts. In the baselines we provide, they appear to work well only for a small subset of genes. But importantly, these are now readouts that you can just get for free given imaging data, given cell painting data in this case. And going in the other direction, we are building models that can predict what a cell may look like if we already know its transcriptional state. There's been some recent really exciting work that's been coming out on this space. And the idea is pretty similar and sort of loosely relates to our previous work where we had synthesized images of cells treated with increasing doses of a compound. This may give us a tool to understand how transcriptional states relate to morphological states, which is something that we can investigate through these simulations. Another application of profiling is to identify gene function by grouping similar genes and alleles. We've tested this idea with genetic perturbations and found an impressive ability to cluster them based on similarity in this work. That's almost 5 years ago, led by Mohammed Roban. When you express each gene in a cell population and cell paint them and extract profiles, you can rediscover in a single experiment all these known biological pathways like RAS, that's up there, and Hippo pathway. Down here, you'll hear more about Hippo coming up soon. We even saw sensible anticorrelations. For example, the negative and positive regulators of the same pathway. We discovered an unexpected such relationship between Hippo and NF-kappa B pathways, which we later confirmed in follow-up work. The next thing we might be able to do, and we don't really have very solid evidence for it so far, but it's to predict drug response for a patient. So a step in that direction is if we can stratify patients and characterize disease alleles based on morphological profiling. Here's an example for lung cancer. Like most diseases, It's not a single disease, it can be caused by a variety of underlying mutations, but we can overexpress each disease-associated allele and look for similarities among their profiles. And we might discover groupings of these alleles into a smaller number of subtypes. The grouping of genes here broadly fits known biology, but more interestingly, when you zoom into a given gene, for example, BRAF, we see that the similarities in the cell painting morphology data mirror what is known about the activity of various alleles. We overexpress the wild type here, and we overexpress each mutant. We see which ones yield similar morphological profiles. And what's really exciting is that you can, it's not a disease-specific assay. You could run it on absolutely every variant uncovered in every sequencing study for every disease to get a handle on the diversity of genetic variants for each disorder. In this same paper led by Juan Casito, we have once again studied the complementarity of cell painting and L1000. Here's a simple way of visualizing the complementarity. Each data point here is an allele that was tested in cell painting. Each data point here is the same set of alleles tested in L1000. And now we have the two plots overlaid with lines connecting the same allele measured in the two assays. All these plots are in CCA space, which is PCA, except that it finds a common subspace for the two assays that are maximally correlated. So most lines are small. This is in most cases, it means that it indicates where the two assays provide similar information, and some lines are long, which is in most cases where the assays provide different information. So it sort of gives you a different view into the complementarity. There's much more to the story. You can read more about it in Juan's paper. There's an exciting technology underway that could really scale up all these experiments. The Blaney lab at the Broad had developed a technology to do pooled image-based screens. And we're now working with them and the Neil lab to create a pooled version of cell painting. The idea is to perturb cells using a pooled perturbation library, for example, a pooled CRISPR library, then do cell painting to get a per cell image-based profiles, and then also read out the barcode of the guide RNA present in each cell. The trick is to first just generate as many copies of the barcode in situ, sequence them in situ by imaging using sequencing. That means you can assign perturbations to each cell as well as have the image-based profile of each cell. It can be a game changer in terms of massively scaling up cell painting genetic screens like the one I just described in the previous slide in the context of lung cancer, but also the rare diseases project where you can now potentially screen hundreds of alleles related to monogenic loss of function disorders. So all of this was laying the groundwork for what is a pretty exciting direction of virtual screening. There are a lot of ways of doing virtual screening, and in this case, we're just using image-based data to do screens. So if you have a gene of interest, you knock it out or you overexpress it. And if you get a really distinctive phenotype, you can go and look up a compound database of cell painting data and see if you find a match. This is sort of exactly analogous to transcriptional profiling using, let's say, the connectivity map, where you're trying to connect genes and compounds using the language of transcription states. So we weren't really sure if it was going to work, but we were really excited to see that it does when we tried it with a very small pilot experiment where we had only 69 genes that met this criteria of a strong phenotype. We tried to match them against a pretty small set of compounds by screening standards, about 30,000 compounds, roughly half of which were distinguishable from negative controls. We looked for compounds that are highly correlated with gene expression, with the gene overexpression, and also compounds that highly anti-correlated with the overexpression. We had known gene compound pairs where it was known that the compound should target the activity of the gene. So we had 63 of those positive controls built into this experiment, and we found roughly 32% of them had the correct compounds at the top of the list. So all that's good, but the proof is really when you actually do a prospective test. So in a prospective test, using genes where we did not know what the answer would be, we grabbed 7 genes, made a list of compounds for each, ran around worldwide really, led by, and Carpenter, our co-leader, contacted several labs. And there were a few that were willing to give it a try. So we had luckily 7 labs willing to take us up on the offer of testing out these compounds because they were experts in those genes and those pathways. 5 of the experiments gave good results. 2 of them fell apart for chemistry reasons, but 3 of them just really worked. In these cases, the top of the list had some compounds that impinged on the pathway that the researchers were studying, which was the expected pathway. One of these was especially exciting. It's a collaboration together with Karen Eisinger at UPenn, focused on hippo pathway regulators. We found 3 really nice regulators, and then we could do a little bit of medicinal chemistry using some analogs that already existed in our library. We tested those, and we found that we could kill sarcoma cells. So these are YAP-dependent sarcoma cell line, and it's really hard to kill. It's a type of cancer that's really hard to treat. So Karen's lab has been thrilled to find these compounds. The blue ones are our original hits, and then we found some even stronger ones that seem to impact this pathway. So we don't know if they bind to YAP1 directly, but they clearly impact this pathway and reduce the growth of this sarcoma cell line. So we are excited to follow these up. And finally, in terms of applications, cell painting can be used to predict other assays. Large-scale assays for drug discovery can be expensive and impractical, particularly for model systems that require primary cells, organoids, or whole organisms. But as an alternative, an international team, including Janssen and many others, had demonstrated that data from a single high-throughput imaging assay can be repurposed to predict the biological activity of compounds in other assays, even those examining unrelated pathways or biological processes. We've extended this in our work, led by Juan Caicedo, Tim Becker, and Nikita Mashkov, together with Kevin Young and from Tommy Yakola and Regina Bosway's group, where we combine chemical structure and transcriptional readouts to predict these assay readouts. The proof of concept results suggest that image data from high content cell assays combined with other information can be leveraged to carefully design compound screens and boost hit rates. Cell painting can be used to predict markers of cell health. Paquita Vasquez and colleagues at the Broad created a cell health assay that reads out a set of 70 or so phenotypes, including the proportion of cells in a specific cell cycle stage, the kind of apoptosis initiated, cell proliferation rates, reactive oxygen species present, and other relevant readouts of cell health. We collected genetic perturbation data across 3 cell lines for a small handful of genes, both in the cell painting assay and the cell health assay. And then we use cell painting features to predict these cell health phenotypes. Greg Way, a postdoc in our group, has shown that we can do a really good job of predicting several of these readouts. You can learn more on GitHub and in the paper itself. So the assay prediction and the virtual screening applications I showed are both data-hungry applications, where the more the data we have, the better. So for that reason, we organized the JUMP Cell Painting Consortium, a group of 10 pharma companies and a few academic groups led by us at the Broad. We've created the largest publicly available cell painting data set. The data set comprises about 146,000 profiles, compound profiles, as well as genetic profiles for a large fraction of the genome, performed across multiple sites. Through this data generation process, we've aimed to address several assay development and computational challenges in cell painting that could further accelerate its use in drug discovery. As of right now, about half the data has been made public. There's a notebook up there on GitHub that shows you how you can access it, but there's a lot more coming up on that front. 2 companies have already created data portals to browse the data, and you can check it out. The data is all hosted thanks to the generosity of the AWS Open Data program, where all the 400 terabytes of this are stored. We think this data set will be a catalyst for machine learning scientists to collaborate on an incredibly rich set of fundamental problems, learning effective representations from images, figuring out how to correct for batch effects, translating signatures across cell types, integrating data modalities, and many more that I'm sure you can think of. One such ongoing effort in our lab is to develop batch effect correction methods, for the most part by just repurposing successful techniques from the single-cell RNA-seq community, where there's been a lot of activity on this front, as some of you might know. Batch effects, especially in high-dimensional biological readouts, can really blur out signal. And our effort here is to come up with strategies that would allow both correcting for batch effects, but more generally integrating data across experiments so that the large data set that we've built can be something that others can easily build upon. Another project using this data is with Pranav Rajpurkar and colleagues at Stanford, who are using language translation models to predict whether a compound targets a gene. They use a subset of the data to train models that essentially predict the likelihood of a gene-compound pair being a true match, given the cell painting profiles of each. Now, predicting the target of a compound is an extremely hard problem, not least because we're dealing with a very, very noisy ground truth. But the team has shown that the methods can beat the method that we'd used in the virtual screening work that I talked about a few minutes ago, although under some heavy constraints, just given the availability of data, because we used a very small subset of the JUMP data set for this experiment, just 300 compounds targeting 160 genes. So we're really excited to test out these models on much larger data sets. Yet another project in this work is led by Robert Van Dyck, who built on ideas by Mekash Babadi at the Broad to learn representations of a sample of cells that can do better than simply aggregating the profiles. The general idea here is that one can use the framework of deep sets in a contrastive setting and then try to learn an embedding of a set of cells such that the replicates of the same perturbation are made to look similar to each other while non-replicates are made to look dissimilar. We found that this method not only improves on the baseline, but can help identify what cells and what features might be driving the phenotype. This model was also trained on a small subset of the data, but the results suggest that the method can be used to learn improved embeddings given enough sample diversity. So with that, I wanted to keep this short so that we could have a discussion around anything I talked about. I'd like to thank all the folks who contributed to this and our funding agencies.
All right. Thanks for the excellent talk, Shantanu. All right. So as usual, if folks in the audience have any questions, please surface them to the Q&A and I will relay that here. And I'll give folks maybe a minute or so to come up with some questions. But in the meantime, Shantanu, I know one kind of area that the single cell perturbational field has started to think about more recently has been causal inference or causal representation learning. And is this something that you and your group have been thinking about in tandem as well?
So I don't know if Juan Caicedo is here, but indeed, Juan Caicedo and Nikita Marshkov, who now leads his own lab, have indeed started framing the problems of representation learning in that setting. There are no methods out there that specifically use causal models per se, but I can drop in a link to Juan's recent paper that sort of provides a general framework for how we can think about these problems, especially in the context of batch effects.
That's fantastic. And so I know you've taken some inspiration from also single cell methods and thinking about batch effects and aleatoric uncertainty. Are there kind of specific methods that you're particularly excited about, or that you think are could be particularly compelling?
Certainly, yeah. So it turns out that the approach that we took was create a baseline based on the existing methods out there, and then build upon it once we have a good benchmarking strategy laid out. So we have all that figured out. It turns out right now, Harmony, which many of you might know, appears to be the best performing on average across the metrics. It still isn't correcting it to the extent that we'd feel confident that the matching would actually provide highly sensitive results, but it is the most promising in terms of its robustness. One downside of it is certainly the fact that you need to reintegrate the data each time. So being able to predict, being able to sort of integrate new data coming in is still a challenge. That's currently something that's handled by Symphony, but that's still only with smaller. You still can do that only with smaller data sets, is what I understand. I haven't fully grokked Symphony at this point, but yeah, short answer, Harmony seems the most promising, but a lot more lies ahead.
Great. So we have a couple of questions that have percolated up in the chat. One question is whether a copy of the slides might be made available.
Certainly, absolutely.
Great, and thank you. And another question from Burton is hot take, single cell versus whole image representations.
Oh, Burton. Hi, Burton. Yes, I'm still betting on single cells, Burton, but I know good reasons for going whole image. That's my hot take.
All right. Any other questions from the audience at this time?
maybe while other people write questions, which we encourage you to do, I have a few questions. That's all right. Yeah. So hi, thank you so much for the talk. My name is Romain, and I have a few questions regarding, I guess, how you aggregate those measurements, and also like how you reason about like the fact that those measurements are single cells. So it's kind of linked to the previous question. The first one, I guess, is when you show like all those effects of like perturbation, for example, you essentially aggregate, and maybe you do not, right? But like one easy way to do is to think about you have all those perturbations, you have all of the cells, and you can kind of look for average effects. Or you could think about effect in distribution across single cells. Exactly. And so either you have a very super strong phenotype, right? Like localization of a protein and all the images look exactly the same. Or maybe you have some kind of a more diverse phenotype, which might happen if you look at like more dynamical type of events, where like there is more stochasticity in differentiation or other stuff. And so in that case, you can have a fraction of the cells in a certain state, another fraction of the cells in another state. The way that this also came up in one of the Paul Blaney's like paper is in the context of optical proof screens, where you can have, like half of the data in a certain cell cycle, the other half of the data is another cell cycle. And what they did in that paper is that they split the data sets and analyze them separately. So I'm just kind of curious as to like how you think about it, and if it's been a huge problem in and I don't know how it works in the cell painting assay, but in that paper I was mentioning, they also have some of the cell painting dyes. So how does it depend on the what you're measuring? How do you think about it in the context of perturbations? And does it contribute to the richness of the phenotype, or is it just some variation you mainly want to take out? And that would be a starting point.
Great questions. In fact, this was the motivation for our long pursuit for figuring out something that can beat the mean as a summary statistic. It has been ridiculously hard to beat. I mean, mean or median, or depending on how you want to sort of aggregate it. And for reasons that are some obvious, some not obvious. It's obvious in the sense that estimation of it is robust. Not obvious in the sense that, even if you are talking about those subtle effects, affecting only subpopulations, it's still the case that as long as you see a consistent such signature, the mean in a high dimensional space is still going to be reproducibly in the same region of the phenotypic space. So even if the mean is not in that sense interpretable, because you're sort of looking at really the synthesis of two subpopulations, as long as you have a reproducible phenotype, that's really all that you care about in terms of the signature. Having said that, it's still unsatisfactory, right? I mean, the fact that you have all this beautiful heterogeneity and you're collapsing it all into one, into the mean is still sort of somewhat, it's just disconcerting. And we've been in the pursuit of methods for that. Back in 2017, 18, lost track, we had a method that was essentially trying to model second order effects, the second order moments of the data, computing the covariance matrix and then reducing it using random projections, and then fusing the second order and first order and such. And that worked, like it worked in the sense that we saw that there's an improved ability to, if you're able to capture the second order moments and then convert that into profile, you're able to get a lift in the curve and the curves that we cared about. The trouble there was that it's still very hard to extend those methods, not very scalable. There's a lot of magic that happens in terms of needing to combine the sets of profiles. So Robert Van Dyck, who's here on the call today, entered with our group and did that work, which I just mentioned, and I believe there's a poster out there as well. And the whole idea there is, can you just learn a representation of this set? And taking the deep set approach, can you simply learn a representation of the set of cells such that it follows some constraints, such that it obeys some constraints and maximizes some objective function, which could be reproducibility, for example. So that appears to, that looks very promising, beats the baseline. Still hard to untangle as to exactly what it's doing, obviously. At first, my hypothesis was that it's learning higher order moments. Still digging into it, but there's good evidence to suggest that you can learn something better than the mean. Robert has done some other simulation experiments showing that. So I guess, long story short, I think that is a very, very promising direction. It may not always show improvements because the mean profile, the mean summary statistic in high dimensions might still be very sensitive, even if you're talking about subpopulations. But I think it's a very valuable direction for research, especially to understand what's happening at the single cell level. I'm sorry, was there any other related question I might have missed, Romain?
No, I think that's mainly it. Yeah, thank you.
Thanks for the question. We have another question that came up during the chat in the interim. So we have a question from Aaron, who asks, can cell painting be combined with spatial transcriptomics?
Great. I don't know if there's anyone from Paul Blaney or JT Neal's lab on the call, but they could probably talk more about the chemistry approach. So they have, the cell painting dyes can be washed off if we do this in a slightly different way. I'll try to drop in a paper where that's being described. So indeed, as long as you can, as long as you can wash it off and continue with whatever else you wanted to do downstream, you're good to go. So in some sense, the pooled optical screening is a sort of much more narrow version of just doing spatial transcriptomics. So the short answer is you can do it. Just need to get the chemistry of the dyes right.
Great. All right. Any additional questions from Shantanu?
I can also ask another question. If do we have some time, Stephen?
Yes, we have quite a bit of time. So
I have a question regarding the. So I think that there is some emerging philosophy that in terms of what we need to measure from those cells, in terms of image profiling, to be able to predict effect of perturbation or understand the phenotype, is only the cell painting dyes. And I might be wrong, but I think from your group or for other groups, there is this excitement that only from those organelles information, you're actually to capture a lot about the biology that maybe is encapsulated in ways that experts cannot really read from the organelles, but that machines can predict. And so I guess I have two sets of questions, but I'll just cut it first to the first one, which is, sometimes you have this specific phenotype you're interested in, for example, aggregation of a certain protein that you think is a model for a disease. So in that case, you decided to measure it and you showed us the slides. And I guess in classical optical screens as well, you can go after a specific protein if that defines a phenotype. But then how do you reason about, do I just need to because at the end of the day, you still want to define that response that still defines your target phenotype, right? So you still need to measure it. But so how do you wrap your head around, should you all cell paint some study specific things we care about? Or if you were to build a huge atlas, for example, of image-based profiling, would you try to do only cell painting dyes and then every expert can have their own case studies in their cells? How would you think about that?
Yeah, well, this is a great question about, what is enough? At what point should you expand the cell painting enough? So for sure, we know that the cell painting dyes are obviously a very small sliver of biology, but at the same time, in terms of structures that you're staining, right? But at the same time, the general idea, I guess, is that you're able to capture a broad set of measurements about a cell by looking at all these aspects of a cell, and to the extent that they are able to reproducibly fall in the same area of the phenotypic space. If you perturb things the same way, then you can sort of trust that they're doing something distinct. Now, the question is whether, now in a practical setting, what should one do when you're sort of studying a disease, but when there's disease biology that tells you to study a specific readout, a specific stain for a specific protein, for example. So we do that, right? So we do that, for example, with cardiomyocytes. We'll hopefully soon have the cardio profiler paper out where we're staining sarcomeres specifically, but there's if you don't stain sarcomeres, there's nothing to study apparently. I mean, almost in a heart cell, and there, take cell painting dyes, drop one, I forget which, swap it out for the for staining the sarcomere, and now you sort of customize the biology a little bit to the thing you care about in this case. It's the same thing with adipocytes. I don't have the slides up here. So there's always a good use case, especially the cell types are very different, to stain for specific things. But the reason why it's always wise to do cell painting is just that to have something consistent, and folks like recursion and in situ know this well. If you want to create a large library that you can accrue upon, you got to keep something fixed. And the cell painting dyes seem to have stood the test of time. So in that sense, they are a way of doing a lookup in a high-dimensional database in a consistent way. But no doubt, if you have specific biology to interrogate, doing it in complement with something else would help you, keep one hook in cell painting and then also be able to investigate the biology. And it might come at the cost of being able to, if you only stain 3 cell painting dyes, that's better than nothing, right? So you can do 3 and then do 1 more of the biology you care about. So that's the strategy we've taken. But the other strategy is just figure out ways of staining more things. The J.T. Neal's lab at the Broad has been working with different technologies that can allow you to sort of stain up 20 different channels. I mean, get 20 different channels readouts by staining it in multiplex. And those are not as scalable, but if the situation demands it, you can do things like that. So you can do both, I guess, is the point.
Got it. Thank you. And I guess my follow-up question is, you mentioned a lot of different cell types. So I guess, maybe the audience will be less interested by this question, but in terms of what we can measure, so after saying what we can measure with the assay, we can discuss about which cells we can profile. So it looks like you actually worked with a diversity of cell types, but there are some cell types that could be very interesting. Like if you think about cells that are not round or that have like NeurIPS or some kind of very irregular shapes, but then you run into technical troubles of, and maybe there, I don't understand the assay perfectly, but maybe there are some technical reasons for which it's difficult to work with those cells in the first place. If you want to CRISPR them, maybe some immune cells are hard to CRISPR. There are a lot of complications to this, for example. But what have you found have you found some main obstacles to, for example, when biologists come to you and say, I want to study this type of cells or this type of biological phenomena, are there some limitations to the assay in what type of cells you can work with and for which reason?
Right. Yeah. So, you can sort of go case by case, but I think we could, for example, you mentioned NeurIPS, right? So we're working with Ronaldo Neme's group at the Broad, at the Stanley Center, to profile NeurIPS. They should be soon. A neuronal cell painting paper coming out soon, where Matt Tegmeyer and others in a group have shown that you can use the same strategy to profile neurons. This is not the only group that's done it. There's one other group in Oxford that I'm aware of, and at MIT who've taken this approach of just doing cell painting on neurons. So it turns out something that I thought would be really complex to do. There's 2 ways of doing it. Image analysis is harder. You may not be able to track every dendron, for example, but you can still sort of get a phenotypic. You can get a phenoprint, so to say, a signature of cell state. The others we could think of are, I guess cells, if they don't stick and grow flat on a dish, that's going to be a problem. But even that, turns out, Spring Discoveries, last preprint that came out, Brandon White and others showed that they had a strategy for doing that with PBMCs. I forget the details. So if there's a will, there's a way, it seems like, with anything, and with cell painting. It's still hard for me to identify one thing that will definitely not work. But in general, we definitely lean towards cells that express their morphology well when they grow in a dish, and things that don't grow in monolayers are painful. Things that are just ball up are hard to use in terms of signatures. But there's a lot that can still be done, apparently.
Oh, I see. Yeah, if the cells also grow in colonies or on top of each other, like epithelial cells, maybe right.
Or they're too smooshed together. Although even with that, if you've seen some of Recursion's papers, their cells are really tightly packed, and that still works, which is probably why Burton talked about whole image profiles.
Yes. Thank you.
Right. Any last questions for Shantanu? Don't see anything in the chat. So if folks don't have any further questions, then I think we can conclude the contributed talk.